Ladies and gentlemen, good afternoon. Thank you for introduction, and I also thank the organizers for the opportunity that I could here speak about my work. My name is AlenaBraunová, and I work in the Institute of Macromolecular Chemistry in Prague, in Czech Republic. It's a very nice city, so I can invite you there. In my lecture, I will deal with block copolymers based on polyhydroxypropyl methacrylamide and polypropylene glycol. We wanted to prepare some amphiphilic block copolymers based on these polymers because we want to gain some copolymers which are not served only as drug delivery system, but always they should inhibit P-glycoprotein to overcome MDR, multidrug resistance. What is it, multidrug resistance? In literature, I found many definitions. What is it? There is some of them. And we can summarize that multidrug resistance is protective reaction of cancer cells which is caused by long-term exposure of cytotoxic drugs on the cells. But how multidrug resistance is developed? One approach said that it is negative effects of chemotherapy because the cancer cells are not homogeneous as healthy normal cells, and some of them are resistant to chemotherapy, a few, but after first chemotherapy, these cells which are resistant to these drugs are not detectable, and they can divide, and the next population from these cells is resulted in a population which is much more resistant to first population of the tumor cells. So the multidrug resistance is very dangerous because the chemotherapy, the next chemotherapy is insufficient, and this is why we wanted to solve this problem. How it works? Multidrug resistant cells try to decrease the drug concentration inside the cells by various mechanisms. One of the mechanisms is drug efflux, when cytotoxic drugs are pumped out from the cell by transmembrane proteins, especially P-glycoprotein. P-glycoprotein is high molecular weight glycoprotein which contain from many amino acids, and this protein is not only in cancer cells, but it is expressed also in healthy cells, in normal cells. It's a member of a large family of ATP binding cassette transporters. They are characterized by these two ATP domains, where ATP is hydrolyzed to release the energy which is required for the transport the drugs out through the cytoplasmatic membrane to extracellular space. So, P-glycoprotein is ATP-dependent efflux pump for xenobiotic compounds with wide substrate specificity. Here you can see that substrates of P-glycoprotein is very different groups of chemotherapy and other agents. As I said, P-glycoprotein is also expressed in healthy cells, but its function is xenobiotic transporter. but there is the expression is not so much not so much amount as the MDR cancer cells, where it is expressed in high levels. So PGP contributes to the cell resistance, to the drug effect, and how to solve it. We can inhibit PGP, but we can inhibit PGP by modifying of drugs, or we can use some inhibitors, special inhibitors of PGP, but there is a risk that the inhibitors are very toxic, and also they could inhibit PGP also in healthy cells. So we can, why inhibit, you can see here, because if we inhibit PGP, we improve penetration of drugs inside the cells, and then the cancer therapy could be more effective. Inhibition of P-glycoprotein is very studied, is very important theme, and for example, a group of Professor Kabanov in USA studied the pluronics as potential inhibitors, and he noticed that the block of polypropylene oxide, polypropylene glycol, works as inhibitor of PGP. It is responsible for PGP inhibition. So we use this information and we design our polymer amphiphilic block copolymer, where hydrophilic part is formed from copolymer of poly-HPMA, polyhydroxypropyl methacrylamide copolymer here, and PPG, polypropylene glycol, which should be responsible for inhibition of P-glycoprotein. On hydrophilic part, on hydrophilic block, we, due to degradable bond, hydrazone bond, attached the doxorubicin, anti-cancer antibiotic, which is one drug on which is substrate, of PGP. Because the physical properties of these two blocks are different, very different, we expected that these block copolymers should form micelle or some nanoparticles, and thus the molecular weight of these systems are increased. So it's very good for passive targeting by enhanced permeability and retention effect. They should be target by passive target to tumor. And covalent binding of doxorubicin, the drug, by pH-sensitive degradable bond is advantage this all these systems are non-toxic, and after delivery to of these systems to tumor, degradable bond should be degraded by pH and the drug is released. So, what is mechanism of inhibition of PGP? Sorry. Low molecular weight drugs by diffusion is enter the cell, and PGP, P-glycoprotein, pump this drug out without any effect. But if the drug is covalently linked to our copolymer, at first P-glycoprotein was inhibited by polypropylene polypropylene glycol, and then the rest of our system can enter the cell, and the drug is released from this system and should work here. Some synthesis of these copolymers. At first, we synthesized the hydrophilic part of the copolymer here by reversible addition, fragmentation, and chain transfer polymerization. Then by polycondensation, we prepare these diblocks or triblocks copolymers if this hydrophilic part is also here. We deprotected this hydrazide group by 3-fluoroacetic acid, and this hydrazide group was then on this hydrazide group was then attached by pH-sensitive hydrazone bond, the drug doxorubicin. The hydrazone bond is really very stable here. This bond is quite fast. And some biological evaluations. Our biological department study in vitro or done in vitro tests. They measure inhibition of cytotoxicity on neuroblastoma cells and on sensitive cells, NB3 without PGP, and on MDR cells, and NB3 dogs, which has PGP overexpressed. As a control, we use low molecular weight doxorubicin and linear doxorubicin. Is doxorubicin which is linked by hydrazone bond to linear poly-HPMA copolymer, so it's polymer doxorubicin. Here you can see that in sensitive cells there are any effect because they have any PGP in their cells, but if we measure inhibition of PGP on MDR cells, you can see that the effect is dramatic. We decreased EC50 10 times. So we can say that our copolymers really inhibit the multi-drug resistance. We also done some in vitro study by calcine assay. There is only polypropylene glycol alone. Here is controls, some low molecular weight inhibitors, and here is diblock polymer precursor. And you can see also that inhibition is quite good. So I can conclude that we synthesize di or triblock copolymers which contained polypropylene glycol as potential PGP inhibitor, and simultaneously drug doxorubicin as an anti-cancer drug. We characterize these systems and study the gradation of hydrazone bond, and biological tests prove that that our copolymers inhibited the PGP. So we expected that our amphiphilic block copolymers are promised for as MDR inhibitors as well as drug delivery systems. In the last, I would like to thank all my colleagues, Ministry of Education, for financial support, and you for your kind attention. Thank you.
One question, if
No, I hope, I think that the P-glycoprotein is the same, but we have systems which are high molecular weight, so we our copolymers will be selected because they are high molecular weight, and we suppose that they enter only tumor cells by EPR effect. Yeah? Yeah.
thank you.